All right, hey, good afternoon, everybody. As everybody's getting settled in, a couple quick announcements. Next week on Wednesday, there's a quiz. There's already a topics guide up for the quiz. It includes stuff that we've talked about already. One thing that is on the topics guide for that quiz, but was not on the topics guide for the quiz a couple of days ago, is a couple of things related to the cancer immunotherapy that I talked about actually before the exam, but were not on the exam, and nor were they on the quiz that you most recently took. But you should review back over the cancer immunotherapy, and in particular, the 2 or 3 topics about that are on the topics guide. If you have questions about that, please, of course, email me. I also want to remind everybody that even though there hasn't really been much or really any discussion going on there, another place that you can post questions like that if you want to is on the discussion board on on Canvas. I will be keeping an eye on that, and that's another good place to post questions. I posted 1 or 2 up there, here and there. But that's another option for you, in addition to emailing me or Mauricio if you have questions about the cancer or anything else that's on that topics guide for the quiz. There may be topics on that quiz that are related to earlier topics, just as much of this stuff is cumulative. There was, for example, there was some stuff about cancer on the most recent quiz, sort of related to cancer and evolution. Any questions about the quiz, the topics? I hear a lot of people sort of talk. Do you have questions that you have about any of that? Yeah, sure. What's up?
Are there 2 quizzes, one on the fever and one on the...
Right. So there are 3 things that you have to worry about for Wednesday. Thing number 1 is the quiz. That has nothing to do with the fever book. Thing number 2 is a homework assignment that is about the book The Fever, which was listed on the syllabus as a required book for you to buy. But the first 3 chapters, since I happen to be I have a relative who works for the publishing company, I was able to get their permission to post the 3 chapters from those 3 chapters from the whole book onto Canvas for all of you. You can't reproduce it further, so that's sort of all in all of you. But that is most of what you would need to answer the homework about that book that is due on Wednesday. That book and that homework material will not be on the quiz that day. It will come up later after we've had a chance to discuss in class. Yeah.
Is the reflection online on Canvas?
So I handed that out on Monday maybe, but there's also up on Canvas. It's very similar to the Exam 1 reflection assignment, but there's a little bit of an added component where you think back about how you studied for Exam 2 versus how you studied for Exam 1, and then what you changed, were those changes helpful for you. But that sheet, in addition to having been handed out, is available on Canvas, I think is its own assignment that you can find on the calendar for Wednesday. I announced in class, and I think it might say at the top of the sheet, that it was due Monday, but then on Canvas, I'd written down Wednesday, and so we're just going to go with Wednesday. That way, if anyone was banking on the way it's written up on Canvas, then that's fine. You can turn it in today or Monday if you want. So those are the 3 things that you all have to worry about on Wednesday. Bringing in your homework about the fever reading, bringing in your exam reflection, and being ready for the quiz, which has topics guide already posted. But won't be about the fever, but is about the other topics. Questions about any of that stuff? Yes?
Wait, so for the fever, do we have to write our answers?
It's, again, submit on Canvas. So it's, so if you go under modules or assignments, it's sort of in multiple places, or on the calendar for that Wednesday, you can find the link to the assignment. There's, again, I think a PDF or Word document up there where you can see in advance the questions. There's like 3 questions, and then there's short essay questions. Then you go into Canvas and say, I want to take this. Canvas calls it a quiz, but to me it's a homework. So you take the quiz and write out your paragraph answers and submit on Canvas. Just like for the most recent homework that was due a couple days ago. Other questions about that? Okay, all right, so for today, the main topic is finishing up about meiosis and talking about various things related to sexual reproduction, as well as differences between biologically male and biologically female individuals. We might touch a little bit on some other sort of conditions that involve, for example, people with two X's and a Y, or one X and two Ys, or an X and no Y, or 3X. And also a little bit about Down syndrome and other chromosomal abnormalities will come up as well in the context of that. Considering that somewhere around 0.5% to 1% of the human population has some sort of sex chromosome abnormality at birth, the binary between male and female is already fuzzy, and the more I learn about the difference between sex and gender, the more I realize I don't understand that as well. And so so for me, I don't have enough qualifications to discuss that, and so we're going to focus on sort of the mostly sort of the biological male-female distinction in this. Sort of in general overview, when a sperm and an egg come together, then we call that fertilization. The sperm and egg themselves are haploid. In mammals, including us, the haploid phase of our life cycle is a microscopic phase, a single cell phase. Some plants have macroscopic haploid phases and macroscopic multicellular diploid phases as well. And even some animals, some invertebrates, have macroscopic haploid and diploid phases. For example, in ants and wasps and bees, the haploid phase of the life cycle is the male of those species, and the diploid phase of the life cycle is represented in the females of those species, which are defined by the size of the germ cells that are produced. So things can get quite complicated quite quickly, but for us as mammals, which is our focus in this class, our haploid phase is microscopic, and then after fertilization, there is a process that we will go into much more detail about at the end of this unit and into Unit 4 about development early on into sort of the basic structures, and then we're going to skip past the sort of later prenatal development and early childhood development into adulthood. There's a whole developmental biology class that I encourage you to consider taking in your junior or senior year if you have the opportunity to do so, that goes into much more detail about all of the amazing processes that go on in human and animal development in general. Any questions about what's up on the slide there? Haploid phase, diploid phase of our life cycle. Okay, so before we talk about mistakes in meiosis and aneuploidy, mutations in evolution, and why sex might be either beneficial or not beneficial for an organism, I just wanted to take a moment and reflect back on the activity that we had last class period about genetic drift. And remind you, first of all, that just like natural selection and sexual selection, which are the two main mechanisms of evolution that we talked about prior to Wednesday, genetic drift is another mechanism for evolution. Unlike natural selection and sexual selection, genetic drift is driven by random chance. That was the whole point of the activity last class period with the coin flips, and people had evolution occurring in their little model populations on paper. We were doing sort of a low-scale computer model, essentially, of a low-tech computer model of small populations with random fluctuations in the allele frequencies. Random evolution. And so unlike natural selection and sexual selection, genetic drift is purely driven by random chance. It is very, very powerful. It can be much, much more powerful than any or nearly any natural selection or sexual selection pressure in a small population. Another way to say that is, in a small population, random chance is almost always the most powerful evolutionary force that that population is experiencing. That doesn't mean that natural selection doesn't come into play as well. So in our small populations, we had more populations settle into homozygous little a in the situation where the big A was harmful than we did homozygous big A. So we saw a mix of natural selection and genetic drift in those populations. But when we compare that to the day before, when we were thinking about large populations, the genetic drift was much more rapid changes in allele frequency, just by random chance. One other thing, actually, that I forgot to mention, but that is that I'm going to post on Canvas next week, is that so there's a regular homework but no quiz the following Wednesday, which I believe is the 15th. And instead of the quiz, there is a bonus homework assignment in which you will do something which is essentially to explore a little bit mathematically about something called the Hardy-Weinberg equilibrium, which is in large populations figuring out, based on allele frequencies, when there's random mating and no mutations and no selective pressure and large enough populations that random chance averages itself out, what frequencies of genotypes you expect to see. That is, since it's a bonus assignment, that is something that you will not be explicitly tested on, but it's a worthwhile thing to do, I think, because it gives you a sense for how allele frequency relates to genotype frequencies. There's a lot of discussion in the book about Hardy-Weinberg equilibrium as well. There's a whole section about it that I encourage you to read, or you can even just on Wikipedia, there's a good article there, unless somebody's gone in and sabotaged it in the last couple hours since I last looked. There's a pretty good article about Hardy-Weinberg equilibrium up on Wikipedia, and so even that would be a good resource to do that homework. There will be a regular homework due also at the same time, but then no quiz on that Wednesday the 15th. But anyway, so in larger populations, you do also have genetic drift, but it is a weaker force. Genetic drift happens more slowly because if you flip 400 coins, you're going to get closer to 50% heads and tails than if you flip 2 coins. 2 coins, you easily, 25% of the time, you can end up with 2 heads. Whereas if you're flipping 400 coins, the probability of them all landing on heads is extremely small. And so genetic drift becomes much less of a factor, but it is a non-zero factor even in larger populations. So having sort of been through the activity on Wednesday, and that sort of quick recap from me, I think some people have emailed me and said that they still feel a little unclear about some of this. So I'm just curious what questions people have about the genetic drift material. Anything? Okay, one question I have for you all to think about. We talked, actually, well, let me back up a little bit. On the quiz, which is graded and posted up on Gradescope right now, there was a question about, in a cancer, why, when you find somebody with an advanced malignant tumor, you go in and then maybe their cancer cells have 3 out of the 20, there are 20 possible oncogenes, and in all of the cancer cells, the same 3 oncogenes the same 3 proto-oncogenes have been mutated to oncogenes. And so the question was, why does that cancer made up of this uniform group where all of the cells have those same 3 proto-oncogenes mutated to oncogenes? If you look through on Gradescope, I gave partial credit if your answer was entirely about the absence of tumor suppressor genes and the fact that tumor suppressor genes increase the rate of mutation. That is absolutely correct, that once cells lose tumor suppressor genes, the rate of mutation goes up. But that doesn't explain why we don't have patches of the cancer where oncogene A and C have been activated, and different patches of the cancer where oncogenes D and F have been activated. If there's random mutation going on, which there is, but if that's the main force behind this sort of activation of oncogenes, then you would expect to see a very heterogeneous, very mixed tumor. In fact, the reason that you see a pretty homogeneous tumor is that what happens is, as the benign growth starts to develop, one cell by chance picks up a gain-of-function mutation in a proto-oncogene. And that one cell, by random chance, that has picked that up, now divides faster than its neighbors. And so weeks later, you come back, and all of the other 9,000 cells that were in that benign growth have died off, and that one cell and its million descendants are all you see, because it has beat out its neighbors. It doesn't help its neighbors pick up those same oncogenes. The tumor cells aren't cooperating with this or anything. They're actually competing with each other. And that's what I mean when I say there's an evolutionary process going on in the tumor, where the tumor cells are kind of competing with each other to grow faster than all of their neighbors. And the ones that are growing faster, the ones that get those gain-of-function mutations in proto-oncogenes, are the ones that weeks later make up basically the entire tumor that you see. Does that make sense? There were a lot of variability in that question. That question, being an essay question, was one of the harder ones on the quiz. So what questions do people have about that idea? Sort of, we call this a cell line, or all of the daughter and granddaughter cells or progeny cells of that one lucky cell from the cell's perspective, although bad from the organism's perspective, that picked up that gain-of-function mutation in an oncogene. Questions about that? Okay. One other thing that I wanted to mention that sort of relates this back to genetic drift in a little bit, or sort of actually compares it to genetic drift. Oh, yeah, sure. Sorry, one, yeah.
Could you kind of touch on how cell or cancer evolution can kind of happen at the organism level, and then how it can happen over generations as well?
Well, it happens within an organism. Cancer evolution happens because the few cells that start dividing faster than the rest of the tumor weeks later have become the entire tumor. They're progeny cells. And they've outcompeted the neighboring tumor cells that were a little slower than them. So that's how it happens within the organism.
Like cancer in terms of hereditary cancer.
Right. So, I mean, the sort of simple hereditary cancers that we've talked about, like our retinoblastoma, those are sort of situations where you have a loss-of-function mutation that's recessive at the cellular level but dominant at the organism level, because random chance catches up with you once you do 10 trillion rounds of mitosis to go from that to that, right? In terms of other hereditary risk factors for cancer, that's a much tougher situation. One thing that kind of relates a little bit to some of the stuff that we're going to talk about a little bit later today. Hereditary risk factors for cancer most of the time aren't as simple as retinoblastoma or BRCA2, where it's a clear loss of function mutation in a gene, and that increases your risk of cancer. One example of a way you could have a hereditary risk factor for cancer that does not guarantee you'll get cancer, but you might have, 3 of my 4 grandparents died of cancer by the time they were 60 years old, and you tell your doctor that, and they're like, okay, well, we need to slather you with sunscreen and make sure you take all your antioxidants and whatever and whatever. What could happen, for example, is rather than a loss of function mutation where one of the 2 BRCA2 genes or whatever is completely non-functional, you could have a version of the BRCA2 gene that mostly works but is a little inefficient. And so then it's not really recessive. Even if you lost the one perfect BRCA2 gene in your genome in some cells, you still have a mostly functional BRCA2 gene in those other cells, but it's going to be more likely to miss the occasional mutation. And so what's going to happen then, so no BRCA2, and maybe your cell goes from having 1 mutation per round of mitosis to having 10 mutations per round of mitosis. A weak BRCA2, maybe you go from an average of 1 mutation per round of mitosis to 1.5 mutations per round of mitosis. And so that doesn't get you to the point where you're like, it's scary high risk of cancer, but it does get you to a point where you're more likely to get cancer than your best friend who happens to have inherited 2 perfectly, typical, highly functional versions of the BRCA2 gene. And so that's sort of how hereditariness comes into play with cancer most of the time, and why we think of it as risk factors rather than guarantees, the way we've sort of been talking about it in the last unit with the sort of simple Mendelian approach that we were taking. Does that?
So like if like say you were to some mutation happen, like kind of like is there a difference between like if a mutation happens, you have some somatic cancer before or after you have children?
So
not like the dominance or recessiveness.
Well, so what it depends. No, what it depends on. So it depends on things that we can't see. So in the problems on the exam, I say something that we can't see, which is I said, I have no risk factors for cancer in my genome. And yet, despite no risk factors for cancer, I got cancer because of this bad luck event. If we know that, which usually we don't, then we can say, okay, that cancer happened in my liver, the liver is far away from where sperm is being produced, and therefore, it's not going to influence my kids. If you don't know about my genetics and all you know is that I got cancer, then you might say I'm more likely than my neighbor who doesn't have cancer to be carrying some risk factors. Maybe I'm not. Maybe I got exceptionally unlucky, as the exam question illustrated. But maybe I got just mostly unlucky but also had some risk factors. And if I'm mostly unlucky but it had some inherited risk factors, then that is a problem for my kids. And so in the exam question, it was I tried to word it carefully to make sure that it was clear that there were no inheritable or inherited risk factors. But if you don't know that, then you would maybe infer that it's likely that I had some because I got this cancer. Does that answer that? Okay. Yeah, other questions about that? Okay, one other thing that I wanted to point out in relation to genetic drift and cancer is that we've talked about randomness a lot. And it's easy to confuse and to sort of think that these random things must be related somehow, but in fact they're actually not. In genetic drift, the randomness that we are thinking about is randomness that happens with meiosis. It is this assortment of genes, this segregation of alleles from my mom and my dad, whatever gene A that I got from my mom, whatever version of that, and gene A that I got from my dad, whatever version I got of that, separating so that any one sperm cell either has my mom's gene A or my dad's gene A, but not both. That's Mendel's principle of segregation. And that is random. If 2 genes are on different chromosomes, they assort completely randomly. If they're on the same chromosome, there's a weighted random assortment depending on how close together they are. That's the sort of linkage stuff. But there's a lot of sort of chance involved in meiosis. And the chance, the probability aspects of meiosis is what causes the probability aspects of genetic drift. Does that make sense? So genetic drift happens because of meiosis. Because I'm randomly selecting alleles. I have 2 alleles and randomly one or the other gets passed on to my kids. That's the source of randomness in genetic drift. In cancer, we're talking about mitosis. And in mitosis, there is no sex in mitosis. It is an asexual reproduction. And there is no segregation of alleles. Random chance comes into play in cancer in the context of random mutations. Random mutations can also happen in meiosis. And that's why there's maybe a couple letters in my DNA that don't match up with either my mom or my dad, because there were a couple mutations there. But the mutation rate across generations is very, very low. as compared to across 10 trillion rounds of mitosis, how many slight mutations there are differing one cell of mine from a different cell of mine. So in cancer, the randomness is mutation. In genetic drift, the randomness comes from the random segregation of alleles in meiosis. And so those are two different sources of randomness. And it's easy to confuse them. There are questions about that? Okay, so now on to sort of mistakes in meiosis in aneuploidy. The aneuploidy is, I think, a term that Dika mentioned very briefly once with the guest lecture that we had a few weeks back, which is just when you have an unusual complement of chromosomes in your cells. So remember, sort of, traditionally speaking, every cell in my body happens to have 22 chromosomes from my mom, 22 chromosomes from my dad, plus one X chromosome from my mom and one Y chromosome from my dad. There's a small chance that I might have actually gotten two Y chromosomes and not know about it. We might talk about that a little bit in a few minutes. But you know, putting aside that low probability, most likely I've got 22 pairs of autosomes, an X and a Y. Aneuploidy, Dika mentioned, happens a lot in different, sometimes in neurons for weird reasons that nobody's been able to figure out. You find neurons that have an extra copy of some chromosomes. That probably means something. We have no idea what it means, and we're just going to ignore it. Pretend I didn't say it. If anybody wants to talk about it more, I'd love to dig into the research, but I honestly have no understanding of it myself right now, and so it would take a while to even figure that out, despite the fact that I have a degree in neuroscience. It's sort of a new emerging thing that I don't know about. I think very few people, if anybody, actually, I'm quite confident to say that nobody has a full understanding of what goes on. There's only been a few papers to illustrate it, and there's growing consensus that it happens, but what it means, I think nobody has really any meaningful clues about. So, but when we're thinking about aneuploidy, when we're thinking about unusual sets of chromosomes in cells or in all of the cells of an organism, the key idea that I want you to be remembering is this idea of dosage. And I mentioned this last time. And last time I talked about the example of a gene called FOXP2. And with FOXP2, there are two alleles. There's a functional allele and a non-functional allele. Incidentally, the function of FOXP2 is that it serves as a regulatory transcription factor, which the active version does, which hopefully you remember from Unit 1 means that it binds to these promoter proximal elements right next to the start of a gene, helps the general transcription factor, also known as the basal transcription factor, come in and find that spot, which then brings RNA polymerase into that spot. And then once all that happens, RNA polymerase goes and copies the genetic sequence to make a messenger RNA, which then gets spliced and capped and tailed and then sent out the nucleus and translated. And if it's got a signal sequence at the front of it, then, or in the middle of it, then it gets brought over to the endoplasmic reticulum and finishes translation there. But anyway, so that's sort of all Unit 1 and Unit 2 sort of stuff that you might want to be thinking about a little bit. But FOXP2 is a regulatory transcription factor. I mentioned last time that there are three possible genotypes. So this is the FF. It's common, and it's what we sort of call normal. This is severe language difficulties. This is dead, doesn't survive to birth. And so this tells us that FOXP2 is not like what we normally think of as recessive dominant the way we were thinking about it last unit. What this tells us is that for FOXP2, amount matters, which is exactly what is the case here in these snapdragons or whatever they are, where amount matters for their color. But in addition to that, you can engineer mice to have this, an extra copy of FOXP2 introduced into their genome. And if you do this to a mouse, then it has extremely severe seizures, structural changes in its brain, and reduced lifespan. A normal mouse will live for 2 to 4 years. These mice live for a couple months. And so again, amount matters, but not only does amount matter, more isn't always better. There's like a right amount that we want to have a normal functioning nervous system. Yeah, sure.
So instead of, let's say if it gets 3 dominant copies, 1 dominant and 2 recessive, will that just...
That would be similar to this, yeah. So what matters is the number of functional alleles is what matters. Okay. Yeah, so if you did... Yeah, so just to sort of fill that idea out, so if you did something like this to a mouse, this would be pretty much normal. So the amount of functional protein being made. So in other words, to say that is no functional protein and you're dead, too little functional protein and you have brain abnormalities, too much functional protein and actually have even worse brain abnormalities in this case. Yeah, sure.
In humans, the heterozygotes are measured based on language difficulty. How do they measure the in mice?
They have other communication abnormalities and all other structural brain abnormalities as well. Yeah, sure.
Would it be possible to add a functional chromosome to DNA sequence?
Potentially. So what you're thinking about is something that's called gene therapy. And one thing, so we talked about before in the cancer immunotherapy unit, we talked about using HIV, taking out the HIV genome and inserting into it other genes that reprogram T cells to specifically recognize and target an individual's own cancer and whatever surface proteins those tumor cells are expressing. Using the same idea, people have tried to take, for example, recessive disorders or other disorders where you've got non-functional protein issues going on. An example of that is cystic fibrosis. Cystic fibrosis is an autosomal recessive disorder. 1 non-functional copy, no phenotype. 2 non-functional copies, and your cells are unable to balance salt and fluid in your lungs. And you have a lot of fluid secretions build up in your lungs, and it makes it hard to breathe. There's some other phenotypes associated with it, but the most devastating phenotype is the inability to regulate salts. So people have tried to engineer viruses that will infect the cells in somebody's lungs and insert a working copy of the cystic fibrosis gene into those cells. There's been some success and some setbacks that are quite complicated, and I'll be happy to talk about with people if they want to. But in theory, yes. In practice, it's hard, is sort of the short answer to that question. But it's a great way to think about treating recessive disorders. Okay, questions about that? Yeah, sure.
I'm wondering, why specifically HIV?
Because it is already the surface proteins of HIV know how to find T cells, stick to T cells, and inject whatever genetic information is there. So if you're trying to engineer T cells, you're going to want to start with a virus host for your genome that finds T cells. I don't remember whether they used influenza or rhinovirus or whatever it was for the cystic fibrosis, but you want something that's going to infect your mucosal epithelial cells in your lungs if you're trying to do so. So you take advantage of what's already evolved in viruses to find viruses that will insert the genetic material into the cells that you need to get it into. Because they're a part of the adaptive immune response, and they're the ones that already look for and can kill cancer cells, and we're just sort of helping them find your cancer in particular. Yeah, sure.
So, they
they have to pick the appropriate virus for the cells that they're trying to change.
Correct.
So like the antivirus, it just happens to have like the right properties to get to the right place.
Yeah, so whatever cells need to be changed, I want to get a virus that's going to infect those cells, and then I'll take out its genome. So instead of reprogramming the cell to become a virus factory, I reprogram the cell to start making the right salt balance or whatever I need to do. Try attacking tumor cells or whatever it is that I want to do. Yeah, that's right. Yeah, sure.
How do they like create origin copies?
Well, so we already know the gene sequence, and so all you really need to do is, that's relatively conceptually. Conceptually, it takes a while to work through all the steps, but it's something that people have worked out quite well. How to make a sequence of DNA of whatever sequence we want is a problem that has many steps to work through. How that would take me about 2 hours to explain, but it's something that we are very good at this point, and it's not a difficult problem. If I know the DNA sequence and I want to make that, that's not a challenge. I'll just sort of leave it at that. Yeah. If you want to talk about the steps, we are happy to spend a little while talking about that. Okay, so in meiosis, there are sometimes mistakes that are made, and one of the most common types of mistakes that results in a visible phenotype is something called non-disjunction. So in normal meiosis, we have our homologous chromosomes find each other in meiosis 1, and then at the end of meiosis 1, they're going to go to different sides. Moms and dads could go together 50%, will go together half the time. Dads will go with dads half the time, and mom with moms half the time. The other half the time, dad's number 1 goes with mom's number 2, and mom's number 1 goes with dad's number 2. That's independent assortment of chromosomes, and why we get independent assortment of genes if they're on different chromosomes. And after that, after meiosis 1, the cell's haploid. But it's haploid with duplicated chromosomes, then it needs to go through meiosis 2, which is really just mitosis again, a round of mitosis to separate the sister chromatids. But biology's sloppy, and sometimes mistakes are made. And one mistake that can be made is you can have both copies of chromosome number 2 go to one side, and then chromosome 1 separates like it's supposed to. And so then what you're left with after the second round of division is 2 gamete cells that have only chromosome number 1. They're missing chromosome number 2. And then another 2 gamete cells that have an extra copy of chromosome number 2. And if this happens, instead of happening for chromosome number 2, if this happens for chromosome number 21, then there's a chance of spontaneous abortion, but there's also a chance that the child will be born. And if the child is born, then we say that they have something called Down syndrome, which is a genetic disease that is caused by and characterized by having an extra copy of chromosome 21 because one of the 2 gametes that resulted in this person had an extra chromosome 21 hiding in it. This causes cardiovascular deficits, neurological deficits, and a few other problems as well. So the question for the next 4 minutes, let's say, is considering what's up on the board, why does an extra copy of chromosome 21 cause any problems at all? Why is it a problem to have extra copies of a chromosome? So let's sort of get together with the group, get out a piece of paper, write your names on it, and so on, and think about why is it a problem to have an extra copy of a chromosome?
Continuing on with biology, the more important subject.
And actually, sorry, there's a second question, which is why is the most common thing that we see born an extra copy of chromosome 21, which actually happens to be smaller than chromosome 22. They were misordered. So Y is, don't worry about that last bit. Y is an extra copy of number 21, the most common genotype born So consider its size and the number of genes it has, and there are fewer genes on chromosome 21 than any other chromosome.
Whatever's going on, like if you have the FF
Hi.
So, with the 23 chromosomes,
Yes.
so you start with like your sex cells. Do they have 23 chromosomes each?
They have 23. So the non-sex cells in my body have 23 from my mom and 23 from my dad. The sex cells in my body have 23 total. Some of, like, I might have chromosome, if one of them grabbed at random, might have chromosome 1 from my mom, chromosome 1 from my mom, it's chromosome 2 from my mom, chromosome 3 from my dad, chromosome 4 from my mom, and so it's going to be one copy of each. And actually, they'll not only be just pure from my mom or dad, but like chromosome 1 from my mom with a little chunk of my dad's chromosome 1 swapped in from the crossing over, chromosome 2 from my mom with another little chunk of my dad's chromosome 2 swapped in from the crossing over, chromosome 3 from my dad with a little chunk of my mom's 3 swapped in, and so on and so forth.
In theory, could you like have like more genetic information from like one parent than like the other?
Well, I have more genetic information in my body from my mom than my dad because my mom gave me an X chromosome, which has a lot more genes on it than the Y chromosome that I got from my dad.
Okay.
In addition to that, there's a little bit of DNA in mitochondria, and so everybody has that, and everybody gets that from their mother.
Okay.
But so for you both as women, you have X from dad, X from mom. So aside from mitochondria, you actually have equal amounts from both.
Okay.
Thank you.
Yeah. Okay, so about 1 minute left to finish up this discussion. Maybe a little less. Okay, so why is an extra copy of chromosome 21 bad? Anyone want to share what their group came up with for why this is problematic? Sure, yeah.
The proteins that chromosome 21 codes for will end up being overexpressed to give more copies of chromosome.
Yeah, so I think FOXP2 is not on chromosome 21, but if it were, we would end up with something like that. But now this is not just for one gene, but for a few hundred genes that are all on chromosome 21. So we have a few hundred genes with too much protein. And so you can sort of think of like a cell like a factory where things are all supposed to work together, and if parts of the factory have too many workers and are going too fast, then there'll be sort of buildups of some material and insufficiencies of other materials going on in the factory, which can be damaging to the cells. What about Y chromosome? So there's another keyword that I underlined. The most common thing we see born is the extra copy of chromosome 21. Why is that the most common thing we see born? Sure.
So when we're looking at the chromosomes, like you said, we have to look at the size. So if there's an extra copy of a larger chromosome, that means that there are going to be several thousand more genes that are being produced at a higher rate, and the protein is being produced faster, which would lead to a higher probability of abortion or death explantation portion or death of the
Yeah.
body, whereas since 21 is so small, there are less excess copies of protein being produced.
Yeah, so a large chromosome, would be, instead of a few hundred, we'd be talking about a few thousand genes that are overproducing protein. And so now our factory is in way worse trouble, where there's way more processes that are going out of step with each other. Questions about that? Yeah, sure.
So then why wouldn't you also have like a similar frequency for mutations and having too many alleles for like 20 and 22? If they're like the same size. Is it just based on like
Why don't we ever see extra copies of 22? Yeah, great question. So that gets to not just numbers, but it actually turns out which genes are there. And there's one or two genes that happen to be on chromosome 22 that your body is extremely sensitive to levels on. And so an extra copy of chromosome 22 happens to always be lethal. Even though it's small and doesn't have much genes, probabilistically you might expect it not to be lethal. But in fact, if there's any one gene that is just like super sensitive to level and can kill you if it's off, then you're never going to see a child born like that. Similarly for chromosome 20 and 19 is a little bit tolerated. 17 and 13 happen to be the next most, 18 and 13 happen to be the next most common because 14, 15, 16, 17, and 20 also have one or two genes on them where one extra copy is just death. And so you don't see that. Yeah, sure.
So when plants have the polyploidy state occur during their cellular division, or
I don't know. But you can but usually when you see it, what you see is a plant that has an extra copy of every chromosome, which at least keeps things in balance. Balance matters, but that's only part of the answer, and the rest of it I don't really know. Okay, so this is all great. More questions about this before we talk about sex chromosomes, because sex chromosomes throw a whole other wrinkle into this issue. Okay, so one other thing, a missing copy of a chromosome is also a huge problem, right? We've got a lot of genes like this situation where we've got, we're short a copy. In fact, humans, there's no chromosome that you can survive with one missing copy. Some animals can, but for humans, even chromosome 21, with its few genes, one missing copy is always death. Which would be the result if this gamete was the one that got fertilized or did the fertilizing. Yeah, that's where we're going. Okay, so assuming half the people in this room approximately have 2 X chromosomes, and half the people in this room approximately have 1 X chromosome, that should lead to a little bit of confusion. So I happen to have 1 X chromosome, and the X chromosome is a big chromosome with like 10,000 genes on it. We're not talking about a small little chromosome that I'm missing an extra copy of. We're talking about one of the biggest, most gene-rich chromosomes in my karyotype, in my nuclei, that I'm short a copy on. So that's a problem. And based on everything I've told you just now, you should expect that I never would have been born. But I'm here, so you can infer that I probably was born at some point. So it turns out that that over evolutionary time, there's something that's quite fortunate for me, which has happened. And that is that the X chromosome, every single gene on the X chromosome, on average, is twice as active as the genes on all of the non-sex chromosomes. So, every X gene is 2 times as active. Which then, that's great, that solves my problem, right? I now have balanced amounts of protein, balanced amounts of messenger RNA being produced off of my 22 non-sex chromosomes and my X chromosome. So I'm happy, problem solved. But then this creates now a problem for half of the population who I don't represent, that wasn't there in the first place for them, but I've made a mess. Well, I haven't, but evolution has made a mess for women now. And the mess that evolution has made for women, then, is that now their X chromosomes are making too much mRNA, too much protein. And so what women have to do, what mammalian females have to do, is in every cell in their body, in early development, there's essentially a coin flip that happens where one or the other X chromosomes gets shut off. And so every biologically female person in this room or on this campus is genetically a mix of cells where their mother's X chromosome were shut down or cells where their father's X chromosome. And so another way to say that is women are patchy or women are stripy. There are zones in a woman's body that have patches of mom's X chromosome active and dad's X shut down, and other patches where they have dad's X chromosome active and mom's X chromosome shut down. This rarely comes up, but there's one case where it does sometimes show up in human women. There's a gene on the X chromosome that's necessary for the production of sweat glands. And so sometimes you will see women who are carriers for this gene, who are heterozygotes for this gene, and there will be patches on their skin that produce sweat glands, sweat glands, and other patches on their skin that produce don't produce sweat glands. Some sort of Freudian slip there. Anyway, one place that this is more classically observed or more commonly sort of cited is calico cats. A calico cat is almost always female. You can get male calico cats if they have an unusual complement of chromosomes where they have 2 X's and a Y, which we're out of time, so we'll talk about next time a little bit. But a calico cat who inherits a black pigment-producing gene on one X and a red pigment-producing gene on another will have patches of black and patches of red on its fur. We're actually going to keep talking about this and then move on into mutations and evolution.